Okay, our stream's live. Great. Let's get started. Welcome, everybody. This is our second version of the Meet the Scientist series with Cerebrum DAO. And we're featuring both projects that we funded and letting the scientists behind those projects and the drug developers behind those projects share what they're working on and the results that they're having, and really featuring that to our community and driving community engagement directly with the people that are doing the research that we funded. And we're also inviting very interesting projects that we are either in discussions with about funding, or we just found and discovered and been fascinated by and want to share it with our community. And so we're alternating pretty much every other episode is going to be either a project funded and then on to interesting projects. And so we're here today. I'm Brian Magierski. I'm founding steward of Cerebrum DAO. And Cerebrum DAO, for those that don't know it, is a global community-funded and governed network organization that seeks to end brain disease of all kind. So we envision a world with only healthy brains, and as long as that journey takes, we will be on it. And we're driven by the community of people that are funding us and participating, because we really believe that getting close to the people who are impacted by these diseases is going to help us advance it and allow their participation in the process too. So that's who we are. We are here to talk about the blood-brain barrier today with Professor Ann Eichmann from Yale and D2B3, a project that she and Manuel Mohr have founded and we've discovered. And with Cerebrum, we've had a focus on brain disease and neuroscience and treating brain diseases as a function of aging. And so we're looking at these upstream effects of aging that could be precursors to diseases, and saying, what if we had interventions to protect us as we age? And one of the pillars of our strategy has been looking at blood-brain barrier and the deterioration of the blood-brain barrier. I know a lot of people have been familiar with the term leaky gut when it comes to our metabolic health. And we're thinking the blood-brain barrier may have similar issues and deterioration that could be leading to accelerations of brain diseases as we age. So this has been a fascinating topic. So I'm eager to dive into it. And let's get started by doing some introductions. And so, Anne, if you don't mind, it would be great if you give a brief background on yourself, your scientific background, and what drew you to study the blood-brain barrier.
Okay, thank you, Brian. Thank you for having us on this webinar. A wonderful opportunity to meet new friends and supporters and to talk about our research. So I am a professor, the Ensign professor of cardiology and professor of cellular and molecular physiology at Yale. I'm a developmental biologist by training. I trained in France with Nicole Ledoirin, who's a famous neuroscientist interested in the development of the peripheral nervous system, who mapped the entire nervous nest and all of its cellular offspring. And while I was with Nicole during my PhD, I discovered endothelial cells, and I've become fascinated with them ever since. There are 60,000 miles of blood vessels in every single human being. They develop organotypic specializations. In the case of the brain, they develop this very tight seal between the blood and the brain called the blood-brain barrier. And this is a complicated structure composed of endothelium, pericytes, and astrocytes. And in my laboratory, we've been using genetically modified mouse models to understand the signaling function of receptor-ligand combinations through targeted deletion approaches. And in the course of these studies, we discovered a particular ligand receptor pathway that controls the integrity of the blood-brain barrier. And the way we found this out is by making genetic knockout mice in which either the receptor or the ligand is inactivated. And we discovered that mice with this mutation develop a leaky blood-brain barrier. And so this was very interesting to see, but we then wanted to have a tool that would specifically interrupt only the interaction of this one ligand and this one receptor. And the necessity to develop this tool came from the observation that in many instances in biology, there is no exclusive partnering between one molecule and another. Both can interact with additional partners. And this was also the case for our ligand and our receptor. And so we developed a monoclonal antibody that specifically blocks the binding of this one ligand to this one receptor, leaving other partners unaffected. And gratifyingly, when we injected this antibody into a normal mouse, we could open the blood-brain barrier. And in this case, because it was an antibody drug, it was a transient effect, and the blood-brain barrier eventually resealed again once the antibody was eliminated from the system. And so this was very satisfying to us scientifically, because it proved that we need one ligand and one receptor to control the blood-brain barrier. But it went far beyond that, because as you already said, there are a lot of health issues affecting the brain, and the blood-brain barrier is a big obstacle in delivering drugs into the diseased brain. And so fast forward, we partnered with Manuel, we patented the antibody, and we created a startup company called D2B3 that we're trying to leverage to really revolutionize the way we can treat brain diseases.
That's fascinating. So we're eager to dive into multiple topics on there. So it's just really super fascinating to understand. Yes, we had this idea of how do you protect the blood-brain barrier, but hadn't really thought about can you selectively open it to deliver payloads.
Yes,
so
exactly.
It's mind-blowing. So really eager to dig into this. But first, I want to let Manuel give an introduction to yourself, your background, and what led you, and how did you and Anne meet? What led you to this opportunity?
Yeah, thank you, Brian. Thank you, Cerebrum DAO. Thanks to the whole community for organizing this. Really looking forward to a great conversation, and also specifically, I think at the end we're going to have some time for community questions. I really look forward to hopefully answering some of those. My name is Manuel Mohr. I'm a scientist by training as well. Did a PhD, grew up and did a master's and PhD in molecular biotechnology back in Europe, where I'm originally born and raised. And during my grad school, started coming to the US on a grad school fellowship that for the first time really got me in touch and excited about neuroscience. So I had this strong molecular biotechnology background, and took that with me over into neuroscience and into matters of the brain and spinal cord and peripheral nervous system later. During grad school, and then afterwards during a postdoc at Stanford University, I sort of expanded on that and really got fascinated by not only the intricacies of how a healthy human brain computes information, but also all the different ways and how this system can fail and ultimately cause diseases. And so I realized at that point that I wanted to have a more direct impact on human health and human well-being. And that's when I left academia and went into industry, and have been in different size startup companies ever since, working right at the forefront of biomedical innovation and drug development. And when my family and I moved to the Yale ecosystem, I became a Blavatnik Entrepreneurial Fellow at Yale University, which really allowed me to spend time digging into all of this phenomenal, innovative, promising science that's being discovered and researched at Yale, and looking at it with a slightly different lens of how can we take this science out of an academic system and accelerate it and translate it over into real impact on human health. That was when I met Anne and the team around D2B3, and I was fascinated by the science. It was very close to my heart. Delivery of drugs into the brain has been something not just me, but really the whole field has been struggling with and has been fascinated with for decades. So here I was staring at a potential solution to that long century-old problem, and I really felt like this was my chance to be part of something really big. And so I worked with Anne and worked with the team to start building the company, and we're really fortunate to have crossed paths with Cerebrum Deo and you, Brian, and we hope to really work with everyone on the team to excel this forward so we can have a true impact.
Amazing, thank you. Yes, this is definitely fascinating. So how about maybe we start with the blood-brain barrier itself, and what actually makes it so unique among tissues. And I mean, you've touched on this issue already, that we're trying to keep it intact and it's there to protect our brain, and as it deteriorates or it gets leaky, pathogens and agents get into the brain that could cause disease and immune responses. But on the flip side, it's a barrier to us actually curing some of these diseases. So yeah, first off, what makes it so unique? And second, are you the first to open up the blood-brain barrier or try to? Have there been other attempts? What hasn't worked, and what gives yours promise?
So thank you. These are great questions. We know from transplantation experiments that were done almost 100 years ago that there is no intrinsic blood-brain barrier capacity in an endothelial cell. This capacity is instructed by the surrounding neuronal cells, astrocytes, pericytes, and neurons. And so the endothelium in the brain gets instructed to develop a particular gene expression repertoire that keeps the barrier tight. And it does so through three major mechanisms. One is the formation of tight junctions. So endothelial cells in the brain hold hands very tightly, and they do so by expressing a protein called Claudin-5 that keeps these junctions tight. The second thing is an entire program of transporters that shuttle nutrients in and out of the brain, and there are more than 50 genes involved in these transporters for sugars, for lipids, for all the nutrients that the brain needs, and also drug efflux proteins that will carry any unwanted toxins or pathogens right back out into the blood. And the third issue that is critical at the blood-brain barrier is that many endothelial cells in peripheral tissues that are specialized for nutrient and metabolite exchanges, such as the lung or the kidney, have porous vesicles or fenestrations that allow substances to go through. And this is forbidden or prohibited in the brain. No transcytotic vesicles are found. And so the really cool thing about our molecular discovery, found this out through single cell sequencing, is that our one ligand, one receptor system controls the entire repertoire of the blood-brain barrier. So using our antibody, we target the three cardinal features of the blood-brain barrier, which is we disrupt the tight junctions so that things can go through between endothelial cells from the blood into the brain. We form transcytotic vesicles, which can carry drugs into the brain. And we decrease expression of drug efflux proteins, such as PGP, which will, again, help drugs get into the brain. So that's why we think we have a relatively non-selective approach to transiently open the blood-brain barrier. And we can let through various types of drugs with a size limitation of around 60 kilodalton, as far as our research has shown right now. Now, there are other approaches to open the blood-brain barrier mechanically. So in the old times, people have used the hyperosmotic substances such as mannitol, and nowadays people use focused ultrasound with microbubbles. And so both of these techniques create a kind of a non-selective disruption of the blood-brain barrier. And the extent of damage that actually does to the endothelium, I think, remains to be assessed carefully through approaches like single-cell sequencing and the like going forward.
Excellent, thank you. So, as a follow-on to that, then, is there anything else that you want to share about the discovery and the experimental turning points that convince you that this receptor was central to maintaining the blood-brain barrier, and how you identified the ligand receptor pair?
Mm-hmm. So our initial discovery was a little bit of a serendipitous one, as often happens in science. And our journey started with a control. Our journey started with a control experiment. So my former postdoc, Kevin Boyer, who is co-founder of D2B3 and now has a permanent position and is a team leader at Ensign in France, he started working on UNC5B, and he used knockdown endothelial cells where he silenced the receptor. And then he wanted to look at the response of these cells to angiogenic growth factors. And as a loading control for his Western blot, he put an antibody against Claudin-5, which is this major tight junction protein I just told you about. And very unexpectedly, we saw that Claudin-5 levels are very much down in the UNC5B knockdown cells. We didn't expect that, but it gave us immediately the idea that this could be a pathway involved in regulating blood-brain barrier tight junctions. And then, so we worked on this on the mice for a while, and then we submitted the paper to a journal, and then the editor said, the reviewer said, we want you to find the ligand for this receptor as well. So that was a tall order because there are several ligands, but we got lucky because of two that we tested in vivo. There was a very clear response, is that only one of them was involved in keeping the barrier tight, and the other one had no effect on brain vessels whatsoever. So this suggested a one ligand, one receptor pathway. And like I told you earlier, the antibody experiment then really nailed this. And so did our single-cell sequencing data, because we could see that there was a symmetrical regulation, a phenocopy, if you wish, of the gene expression repertoire that was regulated by the ligand and by the receptor. So this is how this came about.
Brian, if you allow, I'll
yes.
just chime in here and for the people in the audience that don't have as much of a scientific or specific blood-brain barrier background, I wanted to just sort of use a metaphor in describing what happened here, and that is the blood-brain barrier, as Anne described it, has several mechanisms that are required that the blood-brain barrier is actually as tight as we see in a normal, healthy, young human being. The cells that create this blood-brain barrier and keep it tight, they need to constantly be told to do that. So it's almost like a light switch, but instead of flipping a switch, we have to keep pressing the button for the blood-brain barrier to be on. And as soon as we lift the finger off of that button, the blood-brain barrier loosens up and relaxes by itself. And so what we do is we basically just put a cap over that button and prevent the finger from reaching it, right? So that is what our antibody does. Our antibody sits on top of that button, which is this receptor that Anne talked about, and prevents the finger from pressing that button for some time. During this time now, the blood-brain barrier naturally relaxes. And once the cover is released on this button or this receptor, we can have reestablishment of the blood-brain barrier, and it goes right back to normal.
Oh, that's interesting. Thanks for that, Manuel. That's a good analogy to kind of lock in for the broader community. Do you think there are other ligand receptor pairs that could impact the blood-brain barrier or open it up, or other barriers?
Yeah, so that's a great question, and I should have told you about that earlier, but thanks for giving me another opportunity. So there is a pathway that is known to regulate blood-brain barrier integrity, which is the Wnt signaling pathway. And there are, this is a large pathway. There's at least 24 different WNTs, and there are many receptor combinations. But the one that is responsible for maintaining the blood-brain barrier tight has been identified. And there are two brain WNTs. So the two members of the WNT family are specifically expressed in the brain, so they exist nowhere else in the body. And there is a WNT-related protein called neurin that's also expressed in the brain and in the eye, and regulates the integrity of the cerebellar blood-cerebellar barrier and the blood-retina barrier. Now, these molecules, these ligands, bind to a multi-receptor complex composed of at least 4 receptors, and they're a little bit different between the receptors for Wnt7A and Wnt7B and the receptors for neurin. And this pathway is known to do just what our pathway is doing, and namely it upregulates Claudin-5, it prevents the transcytotic vesicles from forming, and it affects drug efflux proteins as well. And remarkably, what we discovered is that our pathway functions as an enhancer of this Wnt signaling to maintain the blood-brain barrier tight. And because it's a one ligand, one receptor pathway, it's a far easier target to manipulate the blood-brain barrier compared to the Wnt pathway itself.
Brian, you mentioned other barriers. Anne mentioned 2 of them, which is retina and cerebellar barriers, which are relatively similar, right? We're still talking CNS barriers. Now we can take that one step further and see if there's any other similar types of blood tissue barriers in the human body. And the answer is yes. We actually find different blood tissue barriers in other tissues as well. Very prominently, very strongly regulated one is the gut barrier. So that regulates what components of a meal that we ingest we take up, and which components we want to excrete again, and we don't absorb into our bloodstream. This is a very important one, and it's actually one that is similarly to the blood-brain barrier, is very, very important target for drug delivery. Here we're not delivering drugs necessarily to the brain, but we can imagine a pill, an orally administered drug that would need to be absorbed through the gut lining first before it can even reach its target organ. So that's a similarly important barrier that we see in the body that is similarly important to drug delivery and poses a pretty big barrier as well, and is under heavy investigation as we speak.
Interesting. So just wanted to touch base on one other comment back to the brain and your ligand receptor pair. And your button analogy, Manuel. Could it be with aging that the ligand that you've identified is depleted and could be a cause of a leaky blood-brain barrier that could lead to, dementias of some kind? And what other insights would you give us about how does the blood-brain barrier change as we age?
Yeah, that's a great question, and is a topic of investigation by many laboratories, including our own, of course. So what we know from studies from the Whisker lab in Stanford, what they did is they labeled the entire plasma proteome with tracers, such that they could see what types of plasma proteins can get into the brain in young and old mice. And what was observed is there's actually more plasma protein getting into the brain in a young mouse compared to an old mouse, but it gets there through very different pathways. In the old mouse, pericytes, which are part of the support system that instructs blood-brain barrier integrity, fall off, and that leads to the creation of transcytotic vesicles and sort of non-selective uptake of proteins from the plasma into the brain. How is that regulated? The short answer is we don't know yet. It is possible that is blood-derived substances that change with aging that would instruct this permeability increase. And in agreement with that, studies have shown that if you fuse together an old mouse and a young mouse by what's called a parabiosis, so you create circulation between these two animals, the young mouse brain ages and the old mouse brain rejuvenates. So it's possible that some of the ligand comes from, that's responsible for this, comes from the bloodstream. Our experiments also show that our ligand is likely present in the bloodstream. And we know this because we can inject our antibody through the bloodstream. So it must bind to the luminal phase of the blood-brain barrier and exert its action there. And so we're testing currently what is the decrease of the ligand in the aging brain, and when do these things happen. It is, however, also possible that there could be ligands within the brain parenchyma that contribute to this leaky aging phenotype, and inflammatory insults, etc., can open the blood-brain barrier from the brain side as well. So there's likely not a simple answer to your question, but it could be a multi-factorial process that we're dealing with.
Got it.
And
okay.
I could add to that because we have a ligand receptor system that is tunable, we can use the system to both open the blood-brain barrier reversibly. We could also use it to stabilize the blood-brain barrier in aging by making an agonist to our receptor. And there are efforts in the laboratory to do this as we speak.
Got it. Yeah, no, that's exactly where I was going to go with that. Okay. Manuel,
yeah, and it sort of begs the question, right? I'm sure a lot of people in the audience are sort of thinking the same thing right now, is why does biology, evolution, come up with a system that can be intricately regulated like this? Why have a switch in the first place if I want the light on all the time? Right? That's sort of the real question here. Why would I have a way to regulate something like the blood-brain barrier if I assume that this is something that is generally good? And the answer probably lies in something that maybe part of the audience might not be as familiar with, which is the blood-brain barrier in a healthy young human being actually is regulated all the time. And it happens during the day and during the night cycle. During the day, figuratively, or at least historically, evolutionarily, when we're running around outside chasing lions or running away from lions, chasing our next meal, when we're exposed to a lot of potentially dangerous influences, that's when we need a really strong and tight blood-brain barrier to protect us from all of these potentially dangerous things. We could get bitten by a snake, and we want to keep that snake venom out of our brain. Now, during the night, when we are resting, when we're sleeping, that's when the blood-brain barrier can actually relax a little bit. And the hypothesis from many scientists in the field here is during the night, relaxing the blood-brain barrier slightly helps with clearing waste products from the brain that build up during the day and allows us to start a new day fresh with a clean brain, so to say. And we believe that this ability to up and down regulate the blood-brain barrier strength is exactly where we are tapping in with our technology here. In a sense, what we're doing is, if you to continue the analogy from earlier, right? Biology has found a way to dim that light switch up and down a little bit, right? And we're using that same light switch, but we're completely removing the finger that wants to press it, right? So we're not just dimming it up and down slightly, but we're really taking it to the extreme and turning the system off for a moment. But this sort of gives a little bit of an idea of why does this exist in the first place, and also how is it physiologically relevant in a normal system?
Yeah, that's hugely helpful, Manuel. That's obviously sleep is so key to our brain health, and that is when the cleanup's happening. But those toxins do have to get flushed out, and it does beg that question exactly. So dialing into this opening up, and so it's there to protect us during the day. So what molecules actually can pass through the blood-brain barrier that's opened with your antibody, and how long is it open for? And is it, is it dangerous even to consider opening it during the day to deliver one drug payload? Are other things going to get through? So I'm curious, first off, the molecules, and then the duration, and then the safety.
Okay. So we are testing this with as many sort of drug moieties as we can currently in D2B3. We initially used just dextrans of different molecular weights and dyes to see what could go through the blood-brain barrier. And so we could deliver small molecular weight dyes, 1 kilodalton. We could deliver dextrans of 10 and 40 kilodalton into the brain, so we can see them pass into the brain, and we can quantify how much of them passes into the brain. But a 70 kilodalton dextran does not get into the brain. We then tried fluorescent bovine serum albumin, which is a molecular weight of about 60 kilodalton. And we saw this is the cutoff. Bovine serum albumin does not get into the brain, at least not over the periods that we measured. And that's actually a safety factor, because large proteins such as the antibody itself or other toxic viruses that may be present in the bloodstream cannot get into the brain under our antibody inhibition because of this size limitation. And the size limitation likely has to do with the transcytotic vesicles that form and the tight junction loosening that develops under our antibody. So there's an inbound safety feature. Now, what can you deliver in that size range? We try to deliver chemotherapeutics against lethal brain tumors. And so we can deliver at least 3 different kinds of small molecular weight chemotherapeutics. And there's very good efficacy against various brain tumor models. So this is an area of development that we are very much looking forward to, because, as you know, brain tumors is one of the biggest challenges in the oncology field. They have been refractory to pretty much everything, including immunotherapy. So new approaches are urgently needed. And that's one area where we are branching out. We can also deliver antisense oligonucleotides very efficiently into the brain. And I think there's a very big industry effort right now to deliver these small oligonucleotides for conditions such as neurodevelopmental disorders or also neurodegenerative disorders such as Parkinson and Alzheimer, where large macromolecules accumulate within the brain tissue and they can be targeted with these small oligonucleotides. So we believe this is a rich area of investigation, and we are looking for partners that would help us develop this antisense oligonucleotide delivery in various brain targets. And lastly, we can deliver growth factors and FAB fragments of antibodies. So full-size therapeutic antibodies cannot go through the blood-brain barrier using our antibody because of the size limitation. But smaller fragments of these antibodies can be delivered. And so this provides additional therapeutic avenues to target various brain diseases. Did that answer all of your question?
I think another component of the question was, how long does the barrier stay open, as well as does that pose any safety concerns?
Yes.
And the short answer to that is, in mice, we've tested that extensively, and the blood-brain barrier is relaxed for about 6 to 8 hours, roughly. That is a time that would be very much in agreement, actually, with the duration of a normal night's sleep, to continue that analogy. It is also a time, it's also a duration that we believe has great safety profile. It is not too long to be a real issue to the patient. It is at the same time long enough to really deliver enough drug. So if you take a drug today or right now and you open a blood-brain barrier, you can imagine that it takes a little bit of time for that drug to really sort of reach all corners of the brain. And so the 6 to 8 hour window enables exactly that. It allows enough time for that drug to really reach all corners of the brain before it starts closing again. And that already starts talking about safety. So we know from an extensive amount of research that's been done on focused ultrasound, and alluded to it a little bit earlier, we know that opening the blood-brain barrier for longer times, about 12 to 24 hours is what focused ultrasound does, for a even wider molecular range of compounds. In their case, it goes up to about 200 kilodaltons in size, has been tested in the clinic and has generally been found to be relatively safe. To double down on that, we actually know, and this is what makes it so confident in the safety of our particular approach. We have done extensive literature research, and we found that it just so happens that this receptor ligand pair, so this button that we're talking about, has been attempted to be drugged, or has been drugged before in clinical trials in humans for a different set of indications. This was in a completely different setting. They didn't even know about the blood-brain barrier and the effects it might have on that. But they did test a compound that works very similar potentially to ours, and it would very likely also open the blood-brain barrier. And again, they took it into human patients, or into human subjects, and tested it there with very, very good looking safety profiles. So they didn't observe any adverse effects in that cohort. So that gives us great confidence that the safety of our approach will be very good.
Yeah, and we know from mouse studies that the only place where we open a barrier in the body with our antibody is in the brain. Everywhere else, we tested whether there's increased permeability, etc., and did not observe anything. So this is in line with the pathway that I told you about earlier, the Wnt pathway, which is a brain-specific pathway, and sort of reinforces that this is the target in the brain. And so this is targeted to the brain vasculature, the heart of the blood-brain barrier, integrity regulation. And from what Manuel said, in clinical studies in patients with peripheral cancers, has had a very good safety profile in terms of targeting. So we think this is, great.
Yes, thank you very much. And that also covered the other question I had, which was how widely distributed do the molecules get in the brain? But thanks, Manuel, on that. So we have about 7 or 8 minutes left, and I do want to leave room for Q&A, but I did want to ask one more question. Maybe we can touch on this in 1 or 2 minutes. About the opportunities that exist if you're successful. It seems very much like you have a platform opportunity here. And also, as I know, there are a lot of off-patent drugs out there, and natural products and a lot of other things that we might be able to test in combination to see if they're effective, because we've never been able to get them through the blood-brain barrier before. So how do you think about the opportunities commercially for your discovery?
Yeah, you hit the nail on the head, Brian. It is, on the one hand side, it's a huge opportunity because we can deliver a vast majority of all current and potential future drug molecules that we would want to deliver to the brain. At the same time, we need to be, of course, very intentional to pursue the right opportunities at the right time, especially as a startup company in today's environment. We need to be very strategic about this. So you hit the nail on the head. There is actually a large amount of drugs that have been approved in non-brain diseases that could also be effective reaching targets and treating diseases in the brain. So this, for example, Anne mentioned a good example earlier. This includes chemotherapeutic agents. You could imagine we already have relatively good drugs to treat peripheral tumors, say, for example, a lung cancer or a kidney cancer or a breast cancer, with chemotherapeutic agents. Now, at the point where one of these tumors might send metastases, and one of these metastases lands in the brain, we are unfortunately very limited in options. We don't really have a lot of good drugs anymore. The same chemotherapeutic that works on a lung cancer will not work anymore on the brain metastases of that lung cancer. And this is exactly because of the blood-brain barrier. Now, we see a huge opportunity in that space where we can take advantage of a drug that's already approved, has a very well-established safety profile, efficacy profile. The FDA is familiar with that drug and knows what to do with it. And we can take it and lift it into a new indication space. So now we can start treating more patients with the same drug. At the same time, I think there is a huge opportunity beyond already approved drugs, where we can go into new spaces, into new drugs that we haven't even researched yet, we haven't even thought of yet, because of the limitations of the blood-brain barrier. So I think in the near term, we focus on opportunities that are more tangible, that are sort of, I hate that formulation, but low-hanging fruit. But then in the medium to long term, we really, I think, are opening new doors and new opportunities to develop drugs that we couldn't even conceive of today. And I think that's where really the growth path of D2B3 hopefully will take us and will take the field.
Excellent. Thank you very much. So we have about 5 minutes left, and I did want to open it up for Q&A. So if anybody has questions, they can post it to the chat. I believe in our YouTube channel is where we're taking questions. So I'll take this first one here. You mentioned that the ligand receptor interaction also inhibited efflux mechanisms. Is this mechanism also time restricted?
Presumably it is currently under investigation. What we see is that when we give ligand, we dial on Wnt signaling pathway and dial up the blood-brain barrier gene expression program. When we give the antibody, we dial this down very rapidly. So most likely this is not just a transcriptional effect, it's also an effect on protein stability. And we do see that major proteins like Claudin-5 get ubiquitinated and rapidly degraded. And this is likely also the case for the drug efflux pump, PGP. So we're looking at that right now, and thank you. This is an excellent question.
I think I see a few questions around duration of the opening in the chat box. So I think we've alluded to that earlier already, but the duration for which we relax the blood-brain barrier is only a few hours at a time, which gives us a great confidence in the safety, and where we have established that we can redose, so we can repeat this treatment in relatively short succession. So that enables us to again have a very versatile platform for the delivery of many different drugs.
Very good.
Another question I see here is whether it is possible that this ligand is actually depleted in aging and dementia, where the blood-brain barrier is weakened. And I'll let you comment on that.
Yeah, so there are reports in Alzheimer's disease that our ligand is a regulated molecule in this context. Now, whether that means it's responsible for blood-brain barrier weakening in this context remains to be determined. So we're looking at this ourselves using publicly available databases of aging blood and aging tissues. But it is in some instances pretty hard to determine how a ligand gets into the bloodstream, or even to prove that its presence in the bloodstream is the relevant source. It requires lots of genetically modulated mice where you knock out the ligand in one particular tissue. Ours is produced by smooth muscle cells, various. And so understanding which ones of those is the responsible one and whether its regulation in aging and dementia is crucial remains to be seen, but it also remains a possibility.
We just got another very interesting question here, where somebody's asking if we could actually open the blood-brain barrier to clean the brain, similar to what happens when we sleep. And it's a very interesting and intriguing question for us. So there is several indications, several diseases where we believe that a weakening of the blood-brain barrier or relaxation of the blood-brain barrier for a transient amount of time might actually be enough to help treat this disease. Now, how does that work conceptually? It's oftentimes these would be diseases where we have a manifestation in the brain, but we also have a peripheral manifestation. And as we know, our body is actually pretty good at fighting off most diseases and combating most diseases. So the peripheral manifestation might be something that our body is able to handle, yet the agents that help the body handle this peripheral manifestation don't make it to the brain. And just by relaxing that transiently and allowing these helpers, what this could be different agents and different diseases, to get into the brain and have their effect there as well, might actually be enough. And in a similar way to how the question is phrased is in diseases like Alzheimer's disease, for example, where we see the buildup of protein aggregates in the brain. We actually believe that, and there's some literature, it's still pretty early days, I would say, but there's a group of researchers that believe that opening the blood-brain barrier and allowing some of those aggregates, some of these proteins, to enter the bloodstream will allow the body to start fighting it and start reducing those aggregates. So as I said, the research in that field is unfortunately still a little bit early for us as a startup company to focus our entire operations on it. But it's certainly something we closely monitor, and we're very hopeful that there will be more opportunities of our antibody to have to impact patients.
Fascinating. That's awesome. So I do want to wrap as we are coming up on time, but I'll pose it to you if there's anything else that you want to share about D2B3, any calls to action to the community, things that you're looking for from participation, recruiting, funding, you name it. Go ahead and feel free to shout it out.
Thank you for offering the stage. So we're as one can imagine, so we're still building the team. We're building operations. We have a footprint here in the New Haven Yale ecosystem, and are always looking for talented people to join the team in the same way as we are building out our science and operational team. We are also always interested to meet like-minded people as well as investors. So we are currently fundraising for our seed series. If there's interested people in the audience, please find Brian or find me directly to talk to us about that. And really, I think a big shout out goes out to, and this is maybe more of a thank you also, to the academic community. It's tough times for academic research right now, but as a startup company, D2B3 that we always stand on the shoulders of giants, and we wouldn't be here without the great academic research that's being done in all of these fundamental research labs out there in the world. And so we hugely appreciate that work, and we really hope that this will continue to be funded and will continue to produce novel, exciting science that we can then follow up and take into the brain and hopefully into patients soon.
Awesome. Well, we'll be championing you through our community as well, and I know we're eager to look at possible collaborations too. So we'll keep advancing those discussions. Thank you so much, both of you, for sharing this master course on the blood-brain barrier. I've learned, we've had conversations before, but I've learned more today again. So every single time we talk, it uncovers new topics for me too. So thank you very much. And thank you to the audience also for joining us for the Meet the Scientists. And we'll be featuring our project Fission Bio next in a couple of weeks. And I know that's being scheduled right now. And I think we might have some exciting reports about our first milestone coming up.
Thank you very much,
having
Brian.
us.
Thank you, everyone.
Absolutely. Thank you.